All right. Hi, everybody. Welcome to Science Sunday summer section. Happy Father's Day to all of you. This is Father's Day. It's also the first full day of summer. The summer equinox was yesterday. And
an eclipse in Asia yesterday,
not
too.
the equinox, a solstice, excuse me.
Oh,
Summer
right,
solstice.
that's right.
Yeah. And an eclipse in Asia?
Yes.
Okay.
Yeah, an annular eclipse, the ring effect where the moon doesn't quite cover the sun, so you get this brilliant ring.
Right.
I have friends in Asia who were able to experience it.
Wow.
It's amazing.
Wow. And also happy Science Sunday today, too. So
Our favorite holiday.
Right, we're doing this through the summer, just because it's a nice way to keep in touch and involved with everybody, and hope you're there and welcome, or if you see this later because you're doing Father's Day activities, and that's understandable, and I hope you're having a wonderful day. So thanks for showing up, and we're talking today about mitochondrial Eve and Y chromosome Adam and the histories of that, and it's been an interesting topic. To me, it's kind of a statistical necessity that there was some level of commonality in all of our lines. That's basically what evolution teaches you. So it's not some amazing, wonderful, crazy thing, but it is something that's been used by 23andMe and the people that are checking DNA sources for people, and we'll get into that as we talk further. I thought it was important to talk first, Scott, you and I, about just what mitochondria are and how they work and what they're doing in cells and why they're there. They're somewhat interlopers, right?
Yeah.
And actually, the more I looked into that got really fascinating to me. The purpose and the value of mitochondria actually are a major reason why multicellularity and why eukaryotic cells, those with a nucleus, could develop and grow and become hundreds, sometimes thousands of times as big as the prokaryotic cells, the original cells that were life on Earth. And the value of the mitochondria in our system, like I said, they're somewhat of an interloper.
They're squatters. They're all squatters.
They're squatters?
Yeah.
They've actually seemed to have been brought into smaller eukaryotic cells that had a nucleus, but they were their own bacteria that then were brought in, maybe in an attempt to consume them, but they found a place and found a home and actually entered into a very important symbiotic relationship with the cell in which they were subsumed. So without mitochondria, cells could not have gone to the strength and power and the activity levels that they've gotten, because mitochondria are the energy sources of cells nowadays. Multicellularity would have almost for sure have been an impossibility, and so that's a fascinating structure. We'll talk a little bit more about what went on with that.
Eukaryotes have typically many mitochondria in them, right? It's not just one mitochondria, like one nucleus.
In fact, it's developed and it's there based upon how much energy that cell needs. So red blood cells basically are just traveling around through your bloodstream. In humans and mammals and other animals, the red blood cells have no mitochondria at all. They don't need energy. They're being pushed through by the pumping of the heart. Whereas
so the heart muscles
the
need
heart
energy.
muscle needs energy,
Yeah.
and something like 40% of the cytoplasm, the rest of the structure inside the cell, but not the nucleus in a heart cell, is 40% are mitochondria.
Wow, I didn't know that
An
number.
amazing amount of processing of power.
Yeah.
But the ability to take oxygen, which is a very combustible source, and change it into energy that feeds the needs of any cell is there. They are found in sperm cells, and sperm cells, they help wag the tail to send the sperm off on their quest for the egg.
And then they die, and they're not used anymore, because the mitochondria out of sperm are just sort of one-shot wonders.
Right.
And once the fusion happens
With the ova,
they're discarded.
Well, not as much discarded as maybe subsumed. Maybe those are consumed by the cell, or
But they're not passed on genetically.
Exactly. And that's the important value, that in a new individual, in any mammal or species, in a new individual, those mitochondria all have the exact same DNA that the mother had prior to them.
So the mitochondria have their own DNA. That's what you're getting at here.
Right, we talk about 23 chromosomes. We've always been brought up that there's 23 pairs of chromosomes, 46 chromosomes in each of our cells, but actually there's another set of DNA in each of the mitochondria. There may be 1,000 mitochondria in one of our cells, and sometimes way more than that too. But each of them has their own DNA as well, which isn't as complex as the DNA in our nuclear structure, but it just controls the ongoing development,
metabolism of
the mitochondria itself. Right. So pretty fascinating stuff. I did kind of jump into this, and I didn't say thanks to the church for opening up the doors here, and for Steve up in the AV Loft, who's running this all for us. And we thought we'd try to do it a little bit more in a relaxed fashion today, because it's summer and we're relaxed, and not quite that relaxed, Scott, but relaxed. Right. And so the physical space and the AV stuff for us, and just the support for Science Sunday, and the ability to get together and talk about things that are of importance to us in our search for truth and meaning.
Yeah, we know you're out there, science lovers.
Yes. So I wanted to also say that this whole superorganism or multicellularity that's able to come about because mitochondria billions of years ago wedged their way into our hearts, so to speak, into our cells.
And our spleens.
And our spleens, and our liver, and our hair follicles, and everything else, is a major transition in evolutionary history, evolutionary biology. Without that happening, this planet would be a completely different planet.
Yeah, the advanced phenotypes. So the genotype is the collection of all the genes that provide the instructions for the construction of molecules, specifically usually proteins. That's the genotype, of which we have 23 pairs of chromosomes that encode that. But one of the results of the action of the genotype is to create a phenotype, a body type. Well, that concept of a phenotype isn't as relevant when you're talking about single-celled organisms. You know, they just have, mostly they're little bundles of metabolism and structure. But the structure, albeit for eukaryotic single-celled life forms, can be complicated with internal organelles and whatnot. The real benefit of having mitochondria within eukaryotic cells is that it allows for life to scale up beyond the microscopic and create complicated phenotypes. You know, everything from an elephant with its prehensile trunk to sharks and snakes and fish and humans and birds that fly in the air and insects
and plants.
Plants, right? Yeah. So the variety of structural adaptations to life in different environments really only happened, just a few hundred million years ago. I mean, it was like recent. 500
million years ago, you're saying that? Multicellularity,
right?
Ediacaran fossils and things like that, right? 600, 700 million years ago.
And we all carry that legacy inside of every cell in our body that contains a genome. Mm-hmm. The history of life on the planet is recapitulated in every new organism that comes about, and you can trace the lineage not just in the fossil record or in trace elements, and it would take me off on a tangent, but not just in the fossil.
Not that.
Yeah, not just the fossil record, but you can trace the history of life by looking at the molecules, which was an astonishing discovery and something that, for example, Darwin had no inkling about, but it has been probably the most relevant sort of science for figuring out how life adapted and how extant living things have come to be.
The other important thing to say is that it's not only mitochondria that have infused themselves into cells to create multicellularity. the same thing has happened in plants with chloroplasts. Another bacterial type of cell, bacterial type of organism that again has come into plant cells. Most plants have chloroplasts, causing them to be green.
Yeah.
And again, it's another energy provider, an energy source, taking sunlight through this small chloroplast, which isn't really the original cell, but it's an addition into the cell, just like mitochondria are for
Right.
ours. So multicellularity in plants was caused by the addition of chloroplasts
Right.
to early cells as well too. They're the first blue-green algae and things like that that started developing chloroplasts, and that gave them the energy that they could then go on and fuel evolution. Without chloroplasts, plants would not have been able to evolve the levels of multicellularity and identities that they have, and without mitochondria, humans or all animals wouldn't have it too. The other interesting thing, before we go deeply into the mitochondrial Eve and so on, is I wanted to say that there's a interesting thing that mitochondrial cells seem to mutate at a faster rate than DNA cells. And the reason that they do that, I would suspect, is that they don't have as much impact on the success of that organism to survive, right? If your mitochondria cell has
the organelle, not
mutate.
the cell, right?
If the mitochondria organelle
Yeah.
mutates, if the DNA mutates in there slightly, then maybe that mitochondria dies, but there's many others in that cell. So
It's like a natural selection within the cell itself
Right.
for either neutral or beneficial mutations, so that it can happen much more rapidly than right. I see what you're saying. The
It may
population sample is so
Right.
much larger.
it may happen at the same rate even, except that it doesn't have the deleterious effects.
Okay.
And I'm back to using deleterious like I used many times in that other talk I gave. But the deleterious effects,
It's got too many syllables.
right?
Just say bad.
Bad effects.
Yeah.
But basically, then it's a good marker because of the mutations occurring or residing, staying in there more recently, more readily. For example, many mutations in your DNA in your nuclear cell, your regular X and Y chromosomes, all your other chromosomes, and your 23 chromosomes, those have a very good potential of causing that mutation, or that mutation has a potential of causing the creature to not survive, to not be able to reproduce.
Yeah.
Although you don't see that happening as much in mitochondrial DNA. So it's actually more of a marker. I think they say there's something like a 2 to 3% difference in the nuclear DNA changes between us and chimpanzees,
Okay. Yeah.
whereas it's about an 8% difference in the mitochondrial DNA
Between us and
chimpanzees.
yeah.
And again, there's like a 12% difference in the mitochondrial DNA between us and gorillas, implying that each of these groups, each of these mutations, the more mutations they are, the further back in time those two
Lineages,
organisms diverged from each other. So it's another proof that supports the fact that we and chimps and bonobos are much more closely related than we and gorillas.
Yeah. Okay.
So anyway, I thought that was an important point to do. The other reason that mitochondrial DNA seem to stay with the woman is that the egg of a woman has thousands of mitochondria in it, whereas each sperm maybe only has 5 or so, small number of mitochondria just to power it.
The little motor.
So it's subsumed by just the volume of
Yeah.
mitochondria in each of those two cells that are going to unite. The nuclear DNA is the same.
Yeah, it's like the sperm. The only real biological function it serves is as a vessel to carry a set of chromosomes, or one. So a haploid. One in a body in a growing organism, typically you have diploid cell. You've got a pair for every chromosome. You've got an X and a Y, or you've got two X's, and you've got two of your number 1 chromosome, two of your number 2 chromosomes, et cetera, et cetera. But the sex cells, the sperm and the ovum, only have a haploid, one complement of each of the chromosomes instead of the pair. But the whole point of the sperm is just to bring that DNA to the ovum, and then the ovum is the one that's in charge of then dividing and replicating and differentiating and providing the metabolism. And so it's really the factory that builds the living organism.
Right. And needs a lot of energy to do all that.
Right.
That's why it's filled with mitochondria.
Yeah, and so every man gets his mitochondria in his cells from the replication of the mitochondria in his mother's original ovum, and every woman also gets the so both males and females carry the mitochondrial DNA and the mitochondrial organelles that are derived only and exclusively from the mother.
Right.
But then the man never passes that on to subsequent generations.
There have been a few exceptions. They
really?
say sometimes, they say sometimes in vitro fertilization when they're doing different things like that. There's been some level of crossing, or I don't think it's contamination.
Crossing over.
But yeah, there's some level of the male's mitochondrial DNA being passed on to those offspring.
I'd never heard of that. That's pretty amazing.
Yeah. Take a little bit more look into explaining exactly why, but there's a few different types. I think they say the bantam roosters, a special type of one kind of chicken, seems to pass on some of this DNA better or whatever. But
in vitro or naturally?
I think naturally in that case. So there are some examples. You never want to say never in science.
Well, especially biology.
Yeah.
We like hard and thin lines between our categories and our ideas, and that doesn't work so well in biology.
Right. It's more of a physics thing.
Yeah, like every cell in your body has a nucleus. Well, except for the ones that don't.
Right. Well, why don't we show this short video that might help portray some of what we're talking about in a more visual way as well, too, and then we'll get on with some slides you brought and some talk about there. So
all right.
Here we go.
Humans inherit mitochondrial DNA only from the mother. So, in order to pass on empty DNA from generation to generation, the mother must have an unbroken link of female descendants. Unlike nuclear DNA, which undergoes recombination, much of empty DNA is passed from mother to daughter, relatively unchanged. If we take a sample of empty DNA from a population and try to reconstruct their matrilineal ancestry using the very slow mutation rate of empty DNA, we might arrive at a single female common ancestor, a mother whom we can all relate to from our mother's side. This is exactly what Kahn, Stoneking, and Wilson did in 1987, and found out that, in fact, all humans alive today do have a common female ancestor who probably lived about 200,000 years ago in Africa. They named her Mitochondrial Eve. Analogous to this, tracing Y chromosomal DNA, which is only inherited from father to son, points to our common father, Y chromosomal Adam. However, there is no reason to believe that mitochondrial Eve and Y chromosomal Adam lived at the same time. Likewise, they were not the only humans living at the time, but their contemporaries failed to produce a direct matrilineal or patrilineal descent. Also, mitochondrial Eve and Y chromosomal Adam were only the most recent common ancestors. Going back in time, we can even find common ancestors of species all the way back to the first single-celled organisms. What's more, our actual common ancestor following both mother's and father's side probably lived as early as 5,000 to 10,000 years ago. All this shows that we are so closely related, more so than we could ever imagine.
I don't like that hyperbole.
Yeah. You should also mention that people can contact us on chat.
I left my phone at home. So yeah, you can feel free, if you have questions or thoughts, feel free to email them to chat@humanistchat.org. However, I forgot my phone today, so I won't be able to answer your question or forward it along to Mike, and we can both address your question until after the talk, unfortunately. So we're just going to have to entertain ourselves.
Oh, man. All right. This changes everything.
Yeah, I know. I wasn't expecting this, having to interact with just one person. Yeah. All right. So this video had a couple of things about it that I think are misleading. One is this notion of Adam and Eve is exclusive to the genes that reside on, respectively, the Y chromosome and the mitochondrial DNA. Every gene in your body, of which the vast, vast majority do not reside on the Y or in the mitochondria, every single gene you have has an origin story and a lineage. In terms of origins, oftentimes a genome will replicate genes. And part of that is believed to enhance survivability because of redundancy. If you have more than one copy of a gene that tends to make the same protein, then you've got an abundant amount of that protein. But if one of those genes changes, if it's a negative, a bad change, or what was that word you used?
Deleterious.
Deleterious change, then then that copy of the gene might be switched off. But if it's a positive survival advantage, then that may essentially create a new trait in the organism, even though it originated as a copy of a legacy trait. So all of your genome is constantly being shuffled around and subjected to mutations and selective pressure. And the Y chromosome and the mitochondria are just two tiny examples of that. So when this video and others talk about mitochondrial Eve and Y chromosome Adam, do not believe that that means that all of your genes originated in those hypothetical individuals. And even the idea that they're a particular individual is this thing we'll probably clarify in a minute. But just because your Y and your X trace to a time and a statistically likely single individual ancestor does not imply anything about the thousands of other genes and the 22 other chromosomes in your body and where they may have originated and with whom and at what period of time. Now, one thing that all of the studies of not just the Y chromosome and mitochondria, all kinds of convergent evidence indicate that human beings and progenitor species originated in Africa. And the Y chromosome and the mitochondrial DNA bear this out because to this day, Africa still has the greatest amount of human genetic diversity of any place in the world, including the diversity of what are called haplogroups on the Y chromosome and the mitochondrial DNA. So you may be tempted at times to say we're all descended from Adam and Eve, this titular Adam and Eve, but that's not specifically true. As I'll talk about when I start showing the slides, every gene has a different trace through history, and it's just the genes on those two packages of DNA that happen to trace to these statistically relevant people.
Right, right. You were talking about Richard Dawkins' great book of...
yes. It's called The Ancestor's Tale, and it's a great Richard Dawkins book.
The Ancestor's Tale, and it's basically, he models it on the Canterbury Tales, and he talks about a journey like done in the Canterbury Tales, but this is a journey back in time, and each point that we diverge from a common ancestor diverge.
The first molecular model of the genetic exchange that occurs.
Steve, could you turn down the volume on that?
Recombination.
I don't need that to play. Sorry about that. All right, So go ahead.
Thanks. So this Canterbury Tales replica, he tells the story of evolution, and I think he only has about 47 points that bring us back to being similar in our ancestry to cyanobacteria and different things like that too. So yeah, it's an interesting.
47 steps along the history of evolution.
Right.
Yeah, it's a beautiful book, The Ancestor's Tale. I highly recommend it. Mike and I have both read it and thoroughly enjoyed. Well, did you thoroughly enjoy it?
Thoroughly.
Yes.
Yeah.
Okay. So this slide talks a little bit about the fact that this notion of identity by descent, where every gene in your particular genome, which is represented at the bottom of this picture in the middle of the slide, little slices of your genome constitute particular genes or groupings of genes, and every gene can trace its lineage back through single ancestors. So you're probably accustomed to the idea of sexual reproduction, and you have replication and mixing of the genome. You have crossover and various other processes that this little animation is highlighting about the way that genes get transferred from one chromatid to another prior to the creation of the gamete cells, the sex cells. And so that evolution has created all kinds of mechanisms, or has evolved all kinds of mechanisms to selectively and randomly grab pieces of DNA and shuffle them on genomes of an individual so that those show up in a random set in their sex cells. But if you look at the resulting DNA, which is represented again by the little circle and the square at the bottom of that center diagram, every single gene you have still can trace back to either a gene in your mother's genome or a gene in your father's genome. There's no mixing of pieces of genes. Similarly, that same gene in your mother or father will trace back to one and only one of their parents. Consequently, every gene in your body traces a single line of descent back through the history of life. Gene collections are shuffled within individuals, but a given gene is only inherited from one parent. Now, because you have two chromosomes, you may have two different genes that carry out an analogous or even an identical function on either of your chromatid, on either of your pairs of chromosomes. And oftentimes you hear about dominant and recessive characteristics, right? The dominant characteristic, if you have only one copy of a particular gene, then you're going to have an effect physiologically from that one copy. A recessive trait is such that you need, even if you have one copy that might be what could be considered defective, it's not going to have a deleterious effect. So all of that is entailed in the way genes work in terms of their pairing, but again, a given gene will only have come from one parent, and one and only one of your grandparents, and one and only one of your great-grandparents, and so on. So that's a misconception, or an easy thing to overlook when talking about genetic.
Right, and you only get half the genes that your mother possessed, and only half of them ended up in the ovum that made you. So right. half of the cells that she had, half of the genes that she had, aren't passed on to you. That's right. Why you're different than your siblings.
Right.
And the same thing.
And that mixture is random.
Side.
Yeah. For a long time, I've been thinking about what's random. Right. I come from a physics background, so I know about quantum mechanics and statistical randomness and whatnot. And in quantum mechanics, there's no cause. There's no cog in the machine that ticks down on the half-life of an atomic particle, for example. There's no structure that can be identified as doing the counting or selecting the statistical outcomes. It is just purely, completely random, which is a whole other topic.
It's like shuffling cards or
Kind of. But on shuffling cards, there's a mechanical underlying mechanism about why each card ends up where it does. In quantum mechanics, there is no underlying physical mechanisms, including the heat of the sun and the direction the wind's blowing and the pattern of skin cells in your thumbprint that catch a card and maybe don't catch a card. So, in principle, in the macroscopic world, everything has a physical cause, and true randomness is exceptionally rare and typically relegated to processes involving nuclear and particle physics and quantum mechanics. So I had this question about how is it that little bits and pieces of the two chromatids that your mother has in, let's say, her X chromosome, little bits and pieces from the part she inherited from her mother and the part she inherited from her father, how did those pieces get intermixed? Well, it's a random process. And as far as I've been able to learn, it's thermodynamic. So it's a stochastic process having to do with the particular prevalence of chemicals that snip the DNA and then transfer it between strands. It's just from even a microscopic view, though not necessarily a particle physics view, from a microscopic view, it's a thermodynamically and chemically random process due to the concentration of different chemicals and enzymes at different places. So that's why when you inherit a chromosome or 23 chromatids from one of your parents, those chromatids share a random distribution of copies of genes from both your grandparents, your male and female grandparents, that are mixed and matched to create a new combination in a single half of a chromosome or a chromatid that your mother then merges with a similarly processed set of chromosomes from your father.
Yeah, you just think of the processes that cause that and whether there is some underlying factor or force, or you'd imagine that certain parts of the chromosome would be more susceptible to breakage.
That is exactly true.
Yeah.
So there are genes that tend to huddle together that get moved as a set. So it's not, I oversimplified it a bit, of course, because it's biology, and biology is far more complicated than I can imagine. But yeah, there are genes that tend to stick together, and that's a complication that is, you're exactly on point with that.
Yeah. Well, you have some more that you want to talk
right.
about in there, and then I did want to make sure we talk about what really are the limitations on mitochondrial Eve, and what to me, it's somewhat of an unfortunate name.
Yeah.
I don't like.
You shared that with me earlier.
Yeah, it's bothersome to me. I mean, I think of the beauty, like I was expressing earlier, of mitochondria and the whole beautiful structure of meiosis and the formation of randomness and potential inside each sperm and each egg that makes each of us an individual. Right. And then to make this being called in like 1987, this mitochondrial Eve tends to bring back religious overtones to it that bother me in some regards too, and maybe we'll get into that a little bit deeper.
I think we'll talk about that a little bit.
Right.
So here's an example of a genetic tree, right? This is a little different than a family tree because remember, each gene, every copy of every gene in your genome comes from one and only one ancestral parent, grandparent, etc. So this traces a lineage of, let's say, 4 different individuals. Those are the ones closer to the middle or bottom, however you want to say it, of the slide. So we've got 4 living individuals that have all inherited the same gene from the individual at the top of this slide.
And the green is standing for one gene.
Yeah, one gene.
Right.
One gene in an individual's genome. So again, there are 4 individuals at the bottom that have inherited this same gene from some ultimate ancestor. Well, if you'll notice, it may be hard to see on this slide, but on the one furthest away from the time arrow, so I don't know, what is that, the left-hand side of the slide, there's one linkage back to a previous ancestor, and then another linkage from that ancestor to a child of the original. And that's different than the other three along the bottom, which have a more recent linkage, and one populated by potentially more siblings. So what happens if, as we're going through and testing the genomes of different individuals, or not even testing, it's just as evolution occurs and as people and genes are selected for or not, or just random chance, some individuals survive, some have offspring, some don't, etc. What happens if we lose this one individual? Well, if we were to go out and do genetic testing now on this population of 3 living ancestors carrying this gene, the most common, or most recent common ancestor of those individuals is only that dot that goes to their to their parents, and then to one grandparent. So, looking on the right side of the diagram, if we only have these 3 bottom individuals that are surviving in a population, we can only at best trace their genetic heritage back to their most recent common ancestor. And I'll make that clear by taking away the part of the tree that is lost because we lost one individual. We lost the record, or maybe we've just not tested enough to have found that individual, but just the lack of statistical evidence can change where this last or most recent common ancestor resides in the tree. And what that effectively causes is that these are lost to history, these carriers of the gene, and the most recent common ancestor shifts forward in time, and now the time looking back to which we can trace the history of that particular gene is foreshortened. Okay? Now, there's the inverse of this story, which really happened in 2013. There was Mr. Percy living in the United States, and Mr. Percy randomly had his genome tested, and through his, I believe, Y chromosome, it could have been his mitochondrial DNA. A male carries mitochondrial DNA, but he doesn't pass it forward. But I think it was through his Y chromosome, a brand new, what's called haplogroup, a brand new set of mutations in a particular genome sequence common in Y chromosomes, a brand new mutated characteristic was discovered. And when his family tree, not his genetic tree of single steps, but his family tree of male and female pairs was traced back, found that he had ancestors in Cameroon. So scientists went to Cameroon and found a village of people who had this commonly this trait. So in 2013, here's what we did. We took this most recent common ancestor, which was dubbed Y chromosome Adam, which lived about 150,000 years ago, and by finding this one person in Cameroon and his relatives, we were able to graft a whole new genetic variant onto the Y chromosomal tree, and that pushed back in time about 100,000 years additional. It added on to our view of the history of the genes on the Y chromosome. Now, all the genes on the Y chromosome are ancient, but in terms of our statistical ability to ferret out where populations split and look at the rates of mutation and extrapolate back, just by finding this one individual and then his related community, we're able to push back the date of the most recent common ancestor.
Which translates into, if you're talking about who is Y chromosomal Adam, it's a different individual now.
Right.
Because new data came in that,
Right.
that changed this whole scenario of what was expected. We thought that everyone was contained within this first subset group that would only go back a certain number of generations, but it pushed it back, I think,
Like almost doubled it.
almost
yeah.
doubled it in time
Yeah.
because of this one individual was found and was traced back, and they can see when those variations occurred. The other interesting thing is that the variations are pretty easily determined. They happen linearly. They happen according to a sequence.
Right.
So that they aren't like odd things happening, but a haplogroup forms when a mutation occurs.
And then that's passed on.
And then that's passed on.
Right.
So anybody that had a haplogroup that was in the descendant of a haplogroup that had formed, let's say 100,000 years ago, they're still in that.
The beneficiaries of that same mutation.
Right, that will show that that'll be in their Y chromosome or mitochondrial DNA.
Right.
Yet maybe further mutations that occur later will divide them into further
Right.
groups. So you can actually see maybe not a chronological time, but a linear progression to these things. So you can see which one of these occurred in these different times. So if you see that this Cameroonian man had other mutations prior to that, you can actually place it in
Right.
the tree of these things. And I think that's what your next slide is talking about.
Yeah, so this is the haplogroups, one of those $10 science words, and it shows mutations that have derived from earlier mutations that have propagated through the Y chromosomal DNA. And these are used to identify genetically related individuals and populations. But one thing to stress, I think we've said this before, but it bears repeating. This is just looking at the history of some genes on one chromosome, or in the case of mitochondrial DNA, the single chromosome that's in the mitochondrial organelle.
But it's of value to do it from those because they're more consistent. They don't mix.
Right, there's no mixing, so there's only patrilineal and matrilineal descent. So the statistics are much easier, and the computer programming is easier.
Right.
So, but this is only one story of one set of ancestors for one set of your genes among thousands. And every chromosome you have and every DNA molecule along the chromosome contains genes that tell a different story.
Right.
I think I was talking to Mike before we started recording today that one of the stories recounted in the Ancestor's Tale is that the BBC made this television program about people who went back to find their ancestors because of their Y chromosome and whatnot. And Dawkins made the point that those ancestors are legitimate. They really do have ancestors that originated in those populations, etc. But that's only a fraction of their genome. Other parts of their genome that can't be as easily geographically tested because of the mixing may have originated in completely different continents with completely different populations, of different languages and traditions. So being able to trace Y and mitochondrial DNA tells us really valuable and interesting stuff, but it's by no means the full picture. And the other corollary of that is that the titular Adam and Eve, which are more titles, that's why I call them titular, rather than specific individuals, those were not the originators of, or the bearers of every gene that you've inherited subsequently. There were many other human beings alive at the time, and from some of those other human beings, or many of them, you've received others of your genes. But again, because of the exclusive matrilineal and patrilineal tracing we can do for the sex chromosomes and mitochondria, it's just easier to find that tree.
Right. So this is not the mother of all.
No.
Mitochondrial Eve is not the mother of all of us in any
Right.
sense other than others who passed on their portions
Right.
of DNA to us were also involved. They're all our descendants, or they're all our ancestors. So it's important to say, I think, one is that she was not the only woman alive at the time, right? That's something that I've seen some science videos or quote science videos that have a religious perspective that are talking about how science has found that yes, there was an Adam and yes, there was an Eve. And that's one reason I feel it, unfortunate that the titles of mitochondrial Eve and Y chromosome Adam were given to them. It's kind of a interesting point, and it has some corollaries to that, but it's not biblical and it's not scientific to call them Adam and Eve, really.
Yeah, it was
They're
sort
not
of
even
a
a
whimsical
fixed individual over
Right.
time.
Right,
as
like
you
we
showed.
said, 7 years ago they found a man who had Cameroonian ancestry that pushed back the title of Adam by 100,000 years.
Right.
And so it's just that it's a conceptual label more than the name of an individual.
And it's important to say that they were not mates.
Oh, right. Yeah.
And they were
good
not
point.
necessarily contemporaneous at the same time.
Right.
They didn't walk through a garden and make comments about walking snakes or anything like that to
Right.
each other. This is nothing biblical involved in it at all.
Well, they may or may not have seen walking snakes. The evidence for that is not present in the genome. So
Right.
we can say it's we have no data about that one way or another, except there's
Well,
no genetic evidence of
And the
walking
scientific
snakes.
evidence of it would be such a rare thing to have them be even contemporaneous. That
Oh,
they wouldn't have
absolutely.
walked together.
Yeah.
They
Well,
could be hundreds of thousands of years apart. It's just that this is the point where the mitochondrial DNA
Mm-hmm.
had a common point that everyone has it.
And then speaking of haplogroups, this is just, I don't expect anyone to be able to read this. You can find this on Wikipedia. This is a map of the prevalence of different prominent haplogroups and how they tend to be distributed geographically and indigenously. Even the term indigenous starts losing some of its connotations when you talk about hundreds of thousands of years of human migration and interbreeding. But in any case, this shows some prevalencies of people who carry certain mutations in the Y chromosome and where these people tend to hail from ancestrally.
Mm-hmm. Mm-hmm.
And then, like you were saying, here's some interesting things we can say about Adam. The ancestral, most recent common ancestor
Is that an actual picture that you got of him?
Yeah, I dug this out of no, of course. Both ancestral Adam and ancestral Eve, along with all of our much earlier and contemporaneous to them ancestors, were residents of Africa. So we're all of us are African, and the whole that gets into the concept of race, which is not something that's particularly useful from a biological standpoint. But in any case, we can say some things about Adam. That was not his name. Nobody called him that. And there was nothing special about him in terms of being a ruler necessarily. Could have been a ruler, could have been, a fisherman, could have been any number of things. We have no idea.
He had offspring.
We do know that. He had at least one male offspring who successfully reproduced.
Right.
And that reproduction led to another male offspring who successfully reproduced. And recapitulate that thousands of times, and that is his claim to fame. He had a whole line of reproducing male descendants. Mm-hmm. Mm-hmm. So that's about the only thing we can say, other than the fact that he was African. And lived about 250-ish thousand years ago, 275. Like you said, never met Eve. One thing that's interesting, I pointed out in other science talks, is there's no gene for original sin, that causes misbehavior or breaks anything in terms of our brains that's been identified in the genome. Certainly not the Y chromosome. So he was not the bearer of some original sin gene, nor was Eve. And his descendants were just lucky to have, well, I don't know, this may be considered too male-centric or whatever.
Yeah.
But lucky may not be the right term, but providentially, he had a continuous line of male descendants. Mm-hmm. That's it. Something almost exactly the same stuff can be said about Eve, except right now we can only push her back to about 180,000 years ago. So by no means contemporaneous.
All right. But I think, that's worth saying. She never met Y chromosome Adam. She also didn't have a sin gene and meet a talking snake, like I was saying.
Yeah.
Her descendants were lucky, and each of them had at least one daughter who reproduced. And she was not the first woman, nor was she the only human female alive at the time.
And she's not the only woman from whom we've inherited the vast majority of our genes, again, to recapitulate,
Right.
reiterate that point. And here's a map of the mitochondrial haplogroups and their prevalency in different parts of the world. And you can find all kinds of information about this for free online.
I think it's interesting. Go back to that 1 second if you can. The colors in there are kind of interesting. And you look at the age in years, 200,000 to 130,000 years ago, and then 130,000 to 90. And so you can see travels and movement of different species.
Right. There's also evidence in the genome of a bottleneck, a population bottleneck, somewhere around 50,000 years ago. There's speculation about what
went down to like 10,000 people or something.
Breeding
like
pairs,
that?
yeah, possibly. Although there's
could fit in a stadium. The whole population of the Earth could fit inside a basketball arena or something like that.
Yeah,
That's
amazing.
That's amazing. Yeah.
Yeah. And there, talking about Adam and Eve, there was never a first human. It's not like two Homo ergasters or Australopithecines gave birth to a human baby, right? There's this idea of the missing links and all of this. The common parlance and high-level undetailed education has maybe given a lot of people a misconception that there was a leap from one species to another, including in the human lineage. But modern humans, indistinguishable from us, are suspected to have first become prevalent about 200 to 300,000 years ago. But 250,000 years ago or 350,000 years ago, we would still see people. They may just have slightly different features or slightly different physiological capacities to us. But the differences would be slight, although statistically relevant.
I know that's an interesting part, I think, a fascinating part of Dawkins' book about the ancestor tale, as he goes back in time and he looks for what he calls hairpin turns. And it's a point at which that species, let's say us as an example, is going back in time, and each one, if you line them up, let's say matrilineally, which he did, you'd see that each, I think he used a rabbit as an example, so I'll switch and talk about rabbits. Each rabbit looked like its mother before it, and looked very much like its grandmother before it. And each rabbit, you would see no real significant difference if you took rabbit number 137 and compared it to 138 or 2,400 generations back to 2,401. But what you would see is some level of change over the immense period of time that evolution needs to succeed.
Yeah. You compare generation 20,000 to generation 5, and you'll see ear length changes or
color changes,
or
a whole variety of different changes. And then this hairpin turn is where that was a point at which, let's say, a rabbit and another type of hare or a similar creature, they both broke off at that point. And so you would have to go down the generation of rabbits down to this point and then start coming back up to another modern creature too, which has a hairpin turn at that point.
And punctuated equilibrium, right? This idea that I think it was Stephen Jay Gould, another famous popularizer of biological science, espoused that historically there's this idea of catastrophism versus gradualism in evolution. The gradualism is things slowly change, like we said, you go back 10,000 generations and you find different rabbit ear lengths, right? That's very gradual. But there's also been this idea of catastrophism, where some population crisis or environmental change led to a sudden rapid shift in adaptive characteristics.
Yeah, we've talked about that with Darwin's finches, and on the islands of the Galapagos, how lengths of birds' beaks changed in a course of 1 year, basically because the ones that were more adapted to work in a dry season were able to survive, while the other ones that weren't able to feed on the seeds that survived through a drought died off. So there can be some amazing quick changes too.
Right, and usually it's guaranteed to be associated with some environmental shift. Either a new predator is introduced, a new disease is introduced, the climate change, et cetera, and makes these thousands of year-long hairpin turns. right? It's not like they happen one generation to the next necessarily. It could take many generations, but from a geologic and biologically evolutionary perspective, they're extremely rapid.
All right. You got a few more slides?
Just this one.
Okay.
So this is, so there was a statistical analysis. I talked about the fact that every gene has a history. There was a statistical analysis done by the folks referenced on this slide. This is an image from the Ancestor's Tale book. And they looked at many genes across different chromosomes and came up with a sort of lineage, genetic lineage of the family tree, identifying major splits. And this is just a representative diagram showing crossbreeding. That's the ladder crossbars, if you will, evidence of crossbreeding that is indicative or extracted from the genome. And when in time, historically, that crossbreeding would have occurred, you'll notice that crossbreeding tended to happen more frequently soon after a split. Well, that's probably because geographically those populations became separated. If you look at the bottom of the graph, it shows, different nation states or continents and whatnot. And so this is evidence that's not post hoc fit into an idea of evolutionary change in the human genome. This is what the data tells us must be true in order for the signals of mutation and the prevalence of mutation to be true. It's not like somebody said we have to fit this into a tree pattern that comes to a single trunk of common descent. This is just what the data is telling us.
I'm questioning how the data knows that there were those horizontal crossing points, I mean, that seems like potentially a supposition, like you say, that they were close to each other still geographically and culturally, so that there would be more interaction.
So the haplogroup idea is from mitochondrial DNA and Y chromosome. It's analogous to a haplogroup, although I'm not sure it's called a haplogroup. It's just evidence of a mutation that shows up in different populations. And the prevalence of that mutation, like if it's really prevalent, it means it's old and it's had time to spread through the population. So you can make some inferences about when that population had contact with another population in which that mutation might have originated.
But are you saying that those horizontal lines then correspond directly to evidence in the genetic mutation levels, that there was a while where there was this interaction, interbreeding between groups, and then there was a time that there wasn't, and that they can see those and how many there were? I mean, it seems.
I think this is schematic. It's not specifically that there were 5 or 7 interbreeding events. It's schematic in that regard, but it does try to convey the prevalence. So if you look at the bottom of the white colored branch, you'll see that between these aren't my distance glasses. I can't see very well the
Italy and North India.
Yeah. So between the people who were native to Italy and North India, there were more frequent exchanges of genetic material than the people between North India and who eventually ended up in, say, Japan and China and Mexico. Mm-hmm. So it's just the points in time when interbreeding generally occurred schematically, and then the relative number of interbreeding frequencies are.
And you're saying that they're getting that by looking at modern people and seeing the mutations that have still been passed along, or what's inside the DNA structure.
Not just the Y chromosome, and that's the big point. Is not this is supporting evidence for the idea of a single common ancestry that also shows how populations diverge, and it has nothing to do with haplogroups from Y chromosome or the mitochondria.
Yeah, I still have some questions about it. Like, why is one set of that dark and one set light?
I think they explain that in the book.
Yeah.
So it does look like it relates to skin pigmentation. The people on the white side tended to live in northern climates where, they didn't need as much UV protection and needed more vitamin D. So I mean, we can start talking about how pigmentation is an adaptive advantage. but has nothing to do with anything except pigmentation, right?
Yeah.
Speaking of adaptive advantages, I wanted
Okay.
to throw in something quick. This book also talks about, remember this book was first written 20 years ago and updated in...
Oh, yeah, more than that, I think, even, yeah.
And updated in like 2015 or 2016. One of the things that this book talked about was how...
The Ancestor's Tale you're talking about.
Yes, sorry.
Okay.
Was the O, B, and A blood types, and the way that those blood types have passed along their genes in the human population, and how there's a mixture. And one of the speculations in this book was that one of the reasons different blood types have persisted in the human population is that there may occasionally be diseases where the proteins expressed by one blood type or the other is more resistant to a particular disease. And that was, it's like I got hit with a hammer, because we're reading now in news that the COVID-19 is more likely to have, by a, not like 100%, but something like 18% more likely to have an impact on people with A blood type.
With A, huh?
Yeah. So this argument that was made in this book, written 20 or more years ago, about blood types may be diverse in the human population and be selected for in order to help protect from diseases.
Wow.
We're seeing an example of that today.
And you just sent me something about CRISPR too, which I read quickly, but we talked a little bit about CRISPR in our science chat 2 weeks ago, and you just found something that in terms of cancer cells, light, and I'm not sure how they put the light into the, but it acted as a, it sped up the interaction of the CRISPR, which happened in like 30 seconds or something like that. An amazing amount of time to control and do that. I don't know if you have...
Yeah, I don't have any more insight than that right now.
Yeah.
Breaking news I just found this morning. So.
Yeah. All right, well, sorry we didn't have any access to questions, and hope that you sent some in to Scott and we can respond to that as well. It's been an enjoyable day. Thanks again to Steve up there, and thanks to you for coming in. I hope you have a wonderful day. I think it's kind of steamy out there, but if you're a father, happy Father's Day to you, and you may be, in generations from now, you may be the new Y chromosome Adam.
100,000 years from now, you may be the lucky one. All right, thanks.
Thanks a lot.